All right, hello. Since the original class on Thursday had some technical difficulties and I was unable to screen share, I am making a separate recording so that if anyone wants to review the lesson again, hopefully it's more clear what I was trying to say. Okay, so today, I will be going over DNA and its processes. So first of all, if you weren't here last week, because we had technical difficulties last week as well, I have included a link to the previous presentation. The recording is also on the Kid Teach Kid website. So if you want to see me struggling to teach, you can see that there. And first of all, before I begin talking about today's actual lesson content, I want to briefly go over some of the structures of a eukaryotic cell. So eukaryotic means multicellular, and that includes things like plants, animals, and humans. On the other hand, prokaryotic is like the opposite, you could say. Prokaryotic means single-celled, and these include things like bacteria. So we're only taking a look at eukaryotic today because that's the only one that's relevant. First thing I want to take a look at is these black dots over here. If you can see them all around. These are called ribosomes, and they will be relevant because it applies to protein synthesis, which is what we're going to be talking about later. You can also see on this I guess line, squiggly line thing, that's called the rough ER, which stands for endoplasmic reticulum. We're not going to be worrying about what the rough ER actually does, but you can see here that there are also ribosomes just like along the rough ER, which is why it's kind of relevant. The next part I want to talk about is the nucleus, which is this pink thing right here, the nucleus and the nucleolus. It doesn't really matter which one, just the nucleus, So the nucleus is important because that is where DNA is stored. It's also known as the brain of the cell and tells the rest of the cell how to function. And that is why it's important. All the DNA inside your cell will be stored inside the tiny little nucleus. Okay, and the last thing I want to take a look at is the cytoplasm, which is right here. That is basically all of the greenish yellow stuff you see in the background. That's kind of like a watery gel-like substance inside the cell to make sure that the small parts. Oh wait, okay, so these small parts, like all of these floating things, they're called organelles. So in the human body, you have organs, right? And they have specific functions. In a cell, they're called organelles. They're basically organs, but they're much smaller, so that's why they're called organelles. And so the cytoplasm is there, it's inside the cell. And I just want to talk about that because I will talk about some things that will travel through the cytoplasm. So I just don't want you guys to be confused. Okay, the rest of this stuff, if you're curious, feel free to search up online. Frankly, I don't know what all of them do because it's not relevant to today's lesson. Okay, so first of all, we're going to be talking about DNA. So what is DNA? DNA stands for deoxyribonucleic acid. And from that, you can see that is why we abbreviate to just DNA, because if we were to say that every time, it'd be very confusing and honestly it would take a lot of time. It's also hard to spell. Okay, so DNA is the genetic code of all organisms. In the human body, we have billions and trillions of cells. And in each of those cells, we have a lot of DNA. Just for context, or just to give you an idea, if you took all the DNA from just one cell, unwound it all, because in order to pack it all in those cells, they need to wind it super tightly. So if you took out all the DNA from just one cell, unwound it all, held it super straight, I guess, it would be the length of 2 meters, which is 6.6 feet. So if you imagine that we have some billions of cells in our body and each DNA in each cell is 2 meters, that's a lot of DNA. So DNA is common in each organism. So the bases are the same and the overall structure is the same. So this slide is just mostly talking about the structure. First of all, DNA has a double helix structure, which means that it's kind of like a ladder, but winded, like a winded ladder. So if we have an example ladder here, you see the rungs of the ladder and the sides of the ladder. If we twisted it, it would look like DNA, which is this diagram over here. And the strands run anti-parallel to each other, which means that they basically just run in opposite directions. You can see I kind of have an example here. So in math class, you might have learned about parallel lines, or just lines that never meet. Usually don't specify direction, but in this case, these strands are anti-parallel. They never meet, but they go in opposite directions, and that is relevant later. And then the actual like components of DNA are nucleotides. So nucleotides are what actually make up DNA. So nucleotides contain a sugar, a phosphate, and a nitrogen base. And we will learn more about that on the next slide. So moving on. Composition of DNA, which is just nucleotides. Okay, so you can see here, this diagram down here is what I mentioned earlier. We have our phosphate group, our pentose sugar, and our nitrogenous base. Okay, so let's talk about the phosphate and the sugar first. So usually we group those together and we call it a sugar phosphate backbone. If you remember from the previous slide, if I can go back, and just ignore the drawings there. This yellow backbone here, that's all sugar and phosphate. So we call it a sugar phosphate backbone. I'm not going to go too in depth into it because it's complicated and frankly, it doesn't really matter in this case. Just know that it is a sugar phosphate backbone. And the most important part for this lesson, at least, is this blue rectangle right here, which is the nitrogenous base. So in DNA, there are 4 bases: adenine, A, guanine, G, cytosine, C, and thymine, T. You just need to remember like the starting letters because that's how we usually write it. Like if we were to write adenine, guanine, cytosine, thymine every time we were writing out a strand of DNA, that'd take forever. So to help you remember the base pairing, so A pairs with C and C pairs with G. So to help you remember, my 6th grade science teacher actually gave us 2, I don't know what they're called, acronyms? I don't know, tricks. So for A and T, you can think of apple and the tree, or if you have some other thing, it would also help. And for C and G, car in the garage. So these don't really help me. I just kind of remember it somehow, mostly because I see A and T and I think at, like the word at, and then I think, oh, there's 2 left, so those 2 must pair together. But if they help you, that's great. So for example, if we're given this DNA strand, GTA, ACC, GTG, you can see that I have split it up into groups of 3, and each group of 3 is called codons. And this is not only easier to read, because if we're all just a long strand, especially if it's really long, like in this case, it's only 9 bases, so it's not that bad, but if it were really long, it would get very confusing very quickly. So first of all, it's easier to organize and, I guess, understand, but also it will be relevant later during protein synthesis when we talk about that. Okay, so let me use my mouse real quick. All right, so if we were to find the complementary strand, which means the opposite strand, we just need to find the base that pairs with each base from the DNA strand. So DNA has 2 strands. Think of this as the first strand, and this is the second complementary strand, so it pairs together. G pairs with C, so this G would be a C, it would pair with C. The complementary base would be a C. T pairs with A, and A pairs with T. So that is how we would get the complementary strand. Hopefully that makes sense. It's not that difficult once you get the hang of it. You just need to remember which base pairs with which base. And that might take a bit of time to memorize, but once you get it, you'll see this is very easy. Okay, so I'm going to move on to the next slide. Here I had a practice problem. I was, honestly, during the original class, I was unable to screen share, so having a practice problem kind of defeated the purpose. So I'm just going to talk through this, especially since nobody else is here anyway. So remember, C pairs with G, car in the garage, and A pairs with C, apple in the tree. And the practice problem is to complete this strand of DNA by finding like the opposite bases. So I'm just going to live solve it, I guess, if you can call it that. So C pairs with G, so we start out with a G, and then G pairs with C. It's kind of like opposite each other, you know. The commutative property, I don't know. No, I don't think that's it. But anyway, C pairs with G, G pairs with C. Works both ways. And then for this one, T pairs with A pairs with T. We have 2 T's here. C pairs with G, G pairs with C, and A pairs with T. My handwriting is really bad, but I'm writing with a mouse, so please forgive me. I can show you. Okay, so yeah, that is what I just wrote, and that would be the answer because we're finding the complementary bases of each base that we're given. Now on to some random facts. I thought of this while I was writing out like the tricks to remember C and G and A and T, because there's also like tricks to remember this, I guess. This is not relevant to the rest of the class, and honestly, it's a little more complicated. Just if you want to know it, here you go. So within the 4 base pairs, we divide it further into 2 groups. Not further, but we divide it into 2 groups. So A and G are purines, which means they have 2 rings. So the 2 rings thing, you can see it right over here. And that literally just applies to like the chemical structure of these base pairs. Not these base pairs, of these nitrogenous bases. And that is why it's not that relevant to this class, because this goes a little more into chemistry. And frankly, I don't even understand what this means. So I'm just telling you in case you want to know. And a way to remember that A and G are purines is the trick sentence saying acronym, pure as gold. So you can see pure for purines, as A, adenine, and gold, G, guanine. The next one is pyrimidines. You can see that over here. They only have 1 ring. So a way to remember this is cut the pi, and pi is spelled P-Y because pi, pyrimidines, maybe it'll help, maybe it won't. And C and T are cytosine and thymine. So you might be wondering, why do we have a U? Then that is our next topic. So what is RNA? RNA stands for ribonucleic acid, and you can see at the beginning there is no deoxyribonucleic acid, which is what DNA would be, and that is because the sugar in RNA is not deoxygenated. However, that is not relevant and a bit complicated. RNA, the biggest differences, I guess, or the biggest things you should notice is that RNA is single-stranded. You can see in the diagram down here, DNA, 2 strands, RNA only has 1 strand. And in RNA, you have the base uracil instead of thymine, which is why earlier when I was talking about pyrimidines, I said cut the pi, so it's CUT, because uracil is now an RNA. And the other bases all stay the same, so A, C, and G. So this just means if we were to do like RNA, do something with RNA, C and G still pair together, but now A and U pair together. So if we had an A in the original strand, it would turn into a U. And if we had a T in the original strand, it would still turn into A. Because you can think of it as A pairs with U and T, but T is only for DNA and U is only for RNA. So that's a bit tricky to like get the hang of at first. I think I made quite a few mistakes just translating A and T both to U, because it's kind of complicated. And the other bases all stay the same. So it's still the same process if you were to like find the complementary bases. It's just slightly different because now you have a U instead of a T. And the RNA can travel outside the nucleus, or it simply exists outside the nucleus. Earlier, at the very beginning, I said that DNA just like lives inside the nucleus. However, RNA can move around through the cytoplasm to different ribosomes, or it literally just like lives outside of the nucleus. And the most common types of RNA are mRNA, tRNA, and RNA. I'll get more into that in the next slide. There are actually a lot of types of both DNA and RNA. The most common one for DNA is just like DNA itself. There's also miDNA, which stands for mitochondrial DNA. You can search that up if you feel like it. But just a brief explanation, it's DNA that you inherit from your mother because it's like the DNA in mitochondria. And that is too complicated, and I will not be explaining what mitochondria are, but if you're curious, feel free to Google it or something. RNA has a lot of different types. There are so many. I had to search it out for a test once. There was so many. I think there's like 20 or something. But anyway, the most common types and the most relevant ones to today are going to be mRNA, tRNA, and RNA. I guess I can go over the diagram at the bottom real quick. So down here you can see DNA has 2 strands, RNA has 1 strand, I already mentioned that. And we can see that here we have thymine and here we have uracil. So that is kind of like the only difference. Well, the biggest differences that you can see from the diagram. Next slide. Okay, so this slide we're talking about types of RNA. First one, mRNA, which stands for messenger RNA, and it is involved in transcription and translation. It travels to the ribosome for translation. And this is obviously very confusing at the moment, but we will get more into that later. So mRNA, just know that it stands for messenger RNA. And the reason that is because it starts out in the nucleus and then it carries all of this information to the ribosome. And tRNA stands for transfer RNA and is involved in translation, and it brings amino acids. So again, this will make more sense when I actually talk about protein synthesis. But transfer RNA, you can think of it as just like bringing the amino acids. It's just called transfer. I feel like I've heard it called like transport RNA at one point. So I don't know if that's actually true or if I just accidentally made that up in my head. But if that helps you remember it at all, just know that it's actually transfer RNA, not transport RNA. You can think of it as like transporting amino acids. And RNA is the last one, which stands for ribosomal RNA. So these are found, our RNA is found in ribosomes, which are like the tiny black dots I talked about at the beginning. And it is the site of translation, which again, I'll make more sense later. Okay, so just as a quick recap, DNA versus RNA. Where's my pen? Ooh, okay. So DNA is double-stranded, RNA is single-stranded, DNA has thymine, RNA has uracil. DNA always stays inside the nucleus, while RNA can travel outside the nucleus or like literally exist outside the nucleus. Moving on. So third section and last section, the processes of DNA and RNA. So I'm going to be going over DNA and protein synthesis, DNA replication and protein synthesis. So just as a disclaimer before I begin, this is going to be like a very brief overview and just like a simple, I'll just simply be going over like the most basic steps within these two processes because it can get really, really complicated. Okay, so first of all, starting with protein synthesis, and I mentioned this a lot earlier because I was talking about like the types of RNA and it got confusing. So protein synthesis mostly just has two main steps. Again, these are the main steps. There are like a lot of smaller steps in between, but I'm not going to be talking about that. So the first step is from DNA to mRNA. So DNA has two strands. Let me just draw a random example. Okay, that doesn't look too great, but whatever. The DNA has two strands. So these strands will like literally split apart, and mRNA comes in, which at the moment it's just like a flat line. But as the mRNA like goes through, it will get like the complementary bases for that DNA strand. So it might get like a, let's just say it starts from here. Let's say this is an A, this is a G, and this is a C. So this mRNA strand, this is mRNA. I apologize for my handwriting. This mRNA strand, it would like pair with that DNA strand. So we would have this one be U, remember A and U now pair together, C, G. Okay, so then after the mRNA is fully transcribed, the mRNA will then leave the nucleus and go into the cytoplasm. So again, mRNA is created using a strand of DNA as a template. The next part is translation, which is from mRNA to proteins. So you might be wondering, what are proteins? Proteins are a very complex molecule in the human body that is responsible for like a ton of functions. So for example, your hair, your skin, it's all because of proteins. And that is why DNA is like the genetic code of all organisms, because it codes for these proteins, which do everything, kind of. So mRNA leaves the nucleus, travels to a ribosome, the rRNA reads it, and then the tRNA brings the amino acid. So I'm going to try drawing a picture. It's not going to look very great, but oh well. So imagine the, oh, that was bad. Wait a minute. Okay, so imagine the mRNA leaving the nucleus, so it's leaving the nucleus over here and goes to a ribosome. Me struggling to draw. Okay, and then the ribosome is pretty much made up of mostly rRNA. I think 80% of it is made of rRNA, and the rRNA will read the mRNA because something needs to like interpret it. It's like coding, kind of. You need something to like read the code and then like interpret it and do the specific functions. So the RNA reads it, and then it calls for tRNA. So tRNA, I have a diagram on the next slide, so it'll make more sense, but I'm just going to draw it as sticks because I can't draw curvy things or bendy things or anything like that. So I'm going to call this a tRNA. The tRNA. So tRNA, this little black dot up here, that's the amino acid. And tRNA kind of floats around and waits for the RNA to call it. Again, I'm going to be using some random bases as an example. So this is a, wait, yeah, fine. CGA, because I kind of flipped it around. Then the rRNA would read it and call for the complementary bases of this like small codon. And that is why we actually split it into threes, because tRNA comes in threes, like the bases, they're in threes. So that is why we usually split DNA into threes, also for ease of reading. Anyway, so this pairs up with GCU. So whatever corresponding amino acid that is coded by GCU would come in to the ribosome, and then the amino acids would like start linking together. And I have a diagram, so hopefully it'll make more sense. I'll just go over what I just said again. First of all, we start with transcription. So you can see the DNA inside the nucleus, it splits apart, and this is the mRNA. You can see it getting its like complementary bases. Then it travels to the cytoplasm to a ribosome. So this gray splotch in the background, that's a ribosome. I wish they had labeled it, but oh well. So you can see the mRNA is going through the rRNA, and you can see these are the tRNA. They're kind of just like floating around. So you can see that the tRNA, the three bases that they have, are getting paired with the three bases on the mRNA. And these amino acids, so think of it like, it usually occurs in like threes. So we have one tRNA here with one amino acid. Let's just say this is the first one. Another one comes in, these two link together, and then a third one comes in, these two link together, and this one like flies off. This doesn't have an amino acid anymore. Its purpose is finished. And the reason it occurs in threes is so that the link will continuously keep going. If the first one already left, then honestly, it wouldn't be that big of a deal. But just understand that it happens in threes, I believe. Just so we have someone that's always standing there. And this chain, it can get very, very long sometimes. Like this one, fairly short, but it can get very, very, very long. And these chains are called polypeptide chains. And after the chain is completely formed and the mRNA is like finished with the reading and whatever, the polypeptide chain will then fold in a very complicated process and pattern to form a protein. And then the protein will go do cellular functions, I guess. They'll like make up your hair or something. Okay, so for an example, just to give you a better idea of how the protein synthesis process works. Let's say we're giving this DNA strand, so GAT, CG, AGT. For this example, I'm just going to be looking at the first codon because I guess that's like the simplest thing I could do. So first of all, we transcribe it from DNA to mRNA. So again, G pairs with C, A pairs with U, and T pairs with A. That is why we get from GAT to CUA. Same applies for the other codons, but I'm not going to do them. And then the next step is we go from mRNA to tRNA. You might be wondering why we skip rRNA, but RNA would technically be in the middle over here because it's the one that's reading. And it's like the ribosome itself, you know. Okay, so C pairs with G, U pairs with A, and A pairs with U. Okay, so at this point, you might notice something interesting. So the DNA strand and the tRNA strand are essentially the same, except the T and the U are flipped, well, interchanged. So every T becomes a U in the tRNA. And that is actually a correct assumption. So during protein synthesis, it goes through like the transcribing or translating process twice. And that is because we need to get the DNA from the nucleus to that ribosome. And we can't do that without translating twice. So the last step is to turn this tRNA strand into a bunch of amino acids. So every three bases in tRNA codes for one specific amino acid. And you can see a giant chart over here. In this chart, all of these amino acids are abbreviated until just like 3 letters because sometimes they have very long names like tryptophan, leucine, phenylalanine, long names. So GAU, this side is the first letter, so we look at this long row right here. So this is the first letter. Second letter is up top, so the second letter is an A. We look at the third row. I apologize for my circling skills. The third column. And then the third letter is the last one on this side, and it just tells us which one to look at within these boxes. So we're looking for a GAU, which is right here, and that codes for ASB, which I believe is like aspartame or something. So that is how we would get the amino acid chain. Some interesting things, I guess, to notice about this chart is that we have, first of all, a red AUG, which codes for met, or methionine or methionine. I don't know how to pronounce it. I always pronounce it methionine, but I'm not really sure. And this is the start codon. So this will just tell the mRNA, the DNA, whatever, where to start with the amino acid. So this is always going to be the first one. In this case, it's not because I just randomly made up something, but usually it would be in real life. Okay, and then the second thing to notice is that we have these 3 over here, and they all code for stop. And it's exactly what it means. If the RNA reads this in the mRNA, it's just gonna stop calling for the tRNA, and that strand, the polypeptide chain, will be finished. It'll fly out and it'll start folding. So the last thing to notice is that a lot of these amino acids, there are multiple that code for just one amino acid, and that actually has to do with these 3 stop codons. Okay, so because protein synthesis is so complicated, you have to like do the translation and transcription twice, and that could lead to some mistakes while like translating or something. You could put in a wrong base or something. So one of the more serious consequences is if somehow it accidentally turned into a stop codon, so if we had the original mRNA strand was, I don't know, AUA, but somehow the tRNA read that, or the RNA interpreted it wrong, or maybe the mRNA was wrong in the first place, it could turn into UAA, which would be a stop codon. So if this is not the actual end of the polypeptide chain, however, the RNA would see this and would say, hey, stop building the chain, and the chain would fly out and just like start folding. However, because it is not finished, it could lead to some negative side effects. And this is called a mutation. And that is why we have multiple codons that code for just one amino acid. And that is because in case there is a mistake, like if you accidentally turned a UCU into a UCC, they still code for the same thing. So even though there was a mistake, nothing's going to change. And that is called a silent mutation because it's silent, nothing happens. There are around, I think, 500 or maybe it's 200 amino acids in like existence, but only 20 are actually in the human body. The other ones are in different animals, plants, that kind of thing. Okay, so moving on to the next slide. This is a practice that I was trying to have, but that didn't work, obviously. So I'm just going to walk you through it again, starting with the mRNA strand. So we just need to translate this DNA strand into mRNA. So, oh, it appeared. Okay. Again, C pairs with G and A pairs with U, so T still pairs with A. So that is how we would get this mRNA strand. The second part is turning this mRNA to the tRNA, and that would be essentially the same thing as this DNA strand, except all the T's are now U's. And in a test, if it just gave you a DNA strand and directly asked you to find like the amino acid chain, you could completely skip the mRNA step and just turn every T into a U. However, most tests will probably have you turn it into mRNA first, and then they might ask you to turn it into tRNA. So it's just an interesting thing to see that they're essentially the same thing. And lastly, the last step would be to find the amino acid chain. If it appears, oh, there it is. Okay. And again, first letter is here. So first one, C, we look at this row. Second one is G, look at this column. And then the third letter is C, so we look at this tiny one right here. There's CGC. And all of these actually code for arginine, which it might not, but in this case, all of these are the same. So hopefully that makes sense. Okay, next we're going to be talking about DNA replication, which is a very, very complicated process. And frankly, I don't completely understand some of the more like difficult parts of it. So first of all, let's take a look at the helicase, which is this triangle blue thing that looks like an arrow. Okay, so the helicase unzips the DNA. You can see that it's kind of like separating these two strands. And that is because for DNA replication, you're obviously trying to make like two copies of the same strand. So in order to do that, in this case, we separated the two strands, and then this strand, the top one, is made into a complete one, the bottom one is made into a complete one. And the reason it works is because, again, we're just pairing the complementary bases, so it turn into two identical strands. Okay, next one I want to take a look at is the topoisomerase, which is this green ring. Okay, so something to notice in this diagram, we can see that this, like the part over here, like all of this, that the DNA part of that, I guess, is kind of straight. It's flattened. But over here, it's winded again. So from this, you can kind of infer that the topoisomerase is in charge of unwinding the DNA or preventing it from supercoiling. Because if it's all coiled up, it's very difficult to like fill in the bases, you know. Next thing is to take a look at these RNA primers. So RNA primers are this thing right here, and they kind of are like a start flag. You know, if you're in a race, there's like a start, you know, that's kind of like a start. And it tells the DNA polymerase, which is this like box thing right here, DNA polymerase, where to start filling in the DNA. Okay, so the next part, I'm gonna go to the next slide because I actually wrote out the steps because hopefully it'll make more sense. So I already talked about the helicase, topoisomerase, and RNA primers. Now it's time to talk about the leading strand and the lagging strand. So over here, you can see there's a lagging strand and a leading strand. And this is why earlier I said that the strands run anti-parallel together. This is where it comes back. Because they go in opposite directions, they are replicated in different ways. So this DNA polymerase, it can only move in one direction. And let's just take a look at the leading strand. So this DNA polymerase, it can only move in this direction. So for the leading strand, it's fairly simple. DNA comes in, just smoothly fills in all the complementary bases. However, with the lagging strand, it's different because it goes in the opposite direction. So if this was the lagging strand, if it went in this direction, remember the DNA polymerase can only go in this direction. So it would have to work like this, and then it jumps back like this, jumps back, and keeps going like that. So that is why it's a little more complicated. And these small fragments right here, they're called Okazaki fragments. You can see them right here. And after like they're finished replicating, the DNA ligase, which is this blue thing right here, the DNA ligase will come in and just like seal the Okazaki fragments together, and that way it will be a complete strand. I think that's it for like the main functions in DNA replication. There are a lot more complicated like enzymes. All of these like small things that are coming in, they're called enzymes. There are a lot more complicated ones, but I'm just talking about like the most simple and basic ones. Okay, so that is it for this class. Again, I apologize for technical difficulties in the original class. I do have homework. Hopefully it will be posted on the Kitty website. This is optional because really you're just here to learn something new over the summer. Homework is really optional. And if you have any questions, especially since this class was so weird originally, or if you want me to check your work, feel free to email me right there. It's literally my name at gmail.com. So I hope you learned something new today, and I hope this class wasn't too confusing. Thank you for bearing with me.